des liens, l'Université de Montpellier fait des travaux sur la recherche et a des liens en particulier avec l'Institut Pasteur de Tunis. On développe depuis longtemps deux axes de recherche, un axe de recherche relativement médical et épidémiologique, dans le cadre du Centre national de référence de Leishmaniose et de le Centre de ressources biologiques, ce qu'on appelle la banque. Et puis un autre axe dans lequel il y a de la recherche fondamentale, et c'est plus de ça dont je vais vous parler aujourd'hui, même si je finirai sur l'activité de recherche translationnelle. I was asked to speak in English, so I will switch in English. If you prefer, you prefer. So I will give you some introduction, and I will give you some introduction. After I will explain how divergent is Leishmania. I will give you data about genomic plasticity. It was the first topic I worked on when I arrived in Montpellier. I won't give you, I'm not sure it is a good one, because I won't give you due to the time, I won't give you anything on genome editing, despite the fact that it was very important for me in the last years. In addition to genome plasticity, we develop 3 other axes, in particular in molecular and cellular biology. And I will present the data we published under the supervision of Patrick Bastien concerning phase 7. I won't give you a large introduction to tubulin tyrosization, and I won't speak at all about epigenetic, despite the fact that it is the topic of my current PhD. And I will finish by translational research. So, some introduction. So, Leishmania is spread all over the intertropical region in more than 88 countries, including southern France and Tunisia. It's a vector-borne disease that also infects mammals. There are more than 20 species that can infect humans. And it gives a large range of disease from self-healing cutaneous leishmania to visceral leishmaniasis. I want to emphasize the fact that it is a divergent eukaryote. You don't see my... we don't have pointer. Okay, so it's a divergent eukaryote. Here is a eukaryote tree of life for eukaryotes, separated in different kingdoms. And here are the Leishmania. And they are divergent very far from model organisms that includes human, yeast, and Drosophila. All of them are there. But also very divergent from other parasites. And here I present Toxoplasma and Plasmodium. It is a divergent eukaryote, and it has many original features. In particular, the mitochondrial DNA, which is structured in thousands of interlocked DNA rings. So it's very dense. We can see it in GMSA staining. It helps to recognize the parasite for the diagnosis. As regards the genomic organization, there are long polycystonic units of non-functionally related genes. There are almost no RNA polymerase II promoters, and most of the regulation is at the post-transcriptional level. There is no entrance, but a very intense transplicing of a splicing leader at the 5' end of messenger RNA. In terms of mitotic cycle, it's a closed mitosis. We can follow the nuclear envelope all along cell cycle. Here it is a nuclear point at level 8. It helps a lot. That's what I present here. Oops. It helps a lot to follow the cell cycle, just determining the number of kineto and nucleus. It has 2 cell cycles, the one of the nucleus, the one of the kinetoplast. They are synchronized, shifted, and independent. As regards kinetochores, quite a long time ago, in electron microscopy, only 6 electron-dense plaques was observed in Leishmania measure, which is not in adequation with the number of 36, which is a number of chromosome heterologs. There is no canonical kinetochores, and kinetochores remains elusive for a long time. Until Akiyoshi, who was a postdoc in his girl lab, was able to describe kinetoplast. Kinetoplast, it's not the last version. Until Akiyoshi and Gull were able to determine kinetoplast protein in T. brucei. They did a screen in order to reduce the number of candidate genes. They labeled some of candidate genes, and they were following. They wanted to see proteins that localized as dots in the nucleus around the S phase, and then move to opposite poles during NF-phases. They use this protein to do ChiP-seq, and they noticed that they are enriched at centromeres. They did serial immunoprecipitations, and they were able to obtain 20 proteins, then 25, and even others. So we took advantage of these results to determine the centromeres in Leishmania. There were the Kakati proteins discovered in T. brucei has autologs in Leishmania. So we tagged it, and we confirmed that the localization was as expected for a kinetochore protein in the middle of the nucleus in interphase or early S phase, and after moving toward the poles at a telophase. We used this tag protein for ChiP-seq, and we were able to find a single narrow peaks to every heterologs. So we were able to characterize the centromeres, and they were essentially non-repetitive sequence. They display great interchromosomal heterogeneity. We found 2 motifs that were more frequent, and also a high density of retroposon. So sorry, I have to come back because I switch. So another very original features is the mosaic aneuploidy. It concerns the number of chromosomes. Since a long time, we knew that it was not a diploid organisms, but we were quite surprised to find by fish that indeed we don't find 2 dots here. It's a fish experiment. We specifically labeled the chromosome 5, and we didn't see that all cells with 2 dots, but in fact a mixture of cells which had 1 dot, some with 2 dots, and some with 3 dots corresponding to 1, 2, or 3 heterologs. We were able to show that several chromosomes were affected. That was observed in several Leishmania species. It has been confirmed by another technique, by WGS. It was at the population level, but when you see coverage like that, obviously there is aneuploidy, and obviously there is mosaic. It was much more recently confirmed in a collaboration paper by single-cell sequences. The role is not totally determined, but there are some clues that are discussed in those papers. What was the most striking was that when we were focusing in dividing cells, and you remember that mitosis is closed, so this is a dividing cells, we found asymmetrical divisions, and always with an odd number, 1 and 2, or 2 and 3, so 3 or 5. This odd number was suggestive of a defect in the regulation of chromosomal DNA replication, leading to the loss or gain of a chromosome copy, followed by a permissive segregation. So we were suspected that in some cells, no firing occurs, and for homologs, and in other cells, double firing occurs. That's why we did DNA combing. DNA combing allows us to stretch DNA fiber uniformly. So we first perform a dual labeling of asynchronous cell population with a nucleoside. We embed the DNA in a melting plug, and then we comb it with that particular device that we built at the lab, and after we reveal with specific antibodies. This technique allowed to measure the replication fork velocity, the interorigin distance, and to estimate the number of origins. So we were able to estimate that there were around 170 active origins per haploid genome. We found large IODs, high speed, more asynchronous firing as compared to MIVEGEC that was used as reference, but we found no clues for double firing events. This was quite disappointing, and I was disappointing since already 8 years. Anyway, we decided to continue on studying DNA replication, and we decided to set up a high-resolution mapping of the origins using nascent strand DNA strand sequencing. So SNS-seq is really a cutting-edge technique, but it's quite difficult. It's a multi-step process. In the lab, we not only work on Leishmania, we also work in T. brucei, a sister parasite, which is responsible for Nagana disease in cattle and for African trypanosoma in humans. So we worked on T. brucei, both on the procyclic forms of the form which is in the insect, tsetse fly, and also to the bloodstream form. And we were able to highly reduce the background using stranded library. That was a very good idea. It was not my idea, but I discussed with the bioinformaticians at the very beginning of the study. And he said, I don't know anything about nascent strands, but as long as you have something symmetrical, you should be able to keep, to know if your sequencing comes from the positive strand or negative strand. So that's what we did. After we did classical NGS sequencing using 150 per hand sequencing, we use a big Illumina in order to have a good depth, and we use 3 replicates and in 2 sequencing. So here are the data. They are quite difficult to interpret because we see many peaks. But we see alternating red peaks, yellow, red and peaks. In fact, we wanted to take benefit of the fact that we use stranded library. So replication bubbles give rise to 2 opposite strand peaks. And these peaks should be in proximity, but not overlapping. That's what I am presenting here. So we play with the maximum distance of 2 peaks, and we also play with the maximum overlaps. So we were able to determine criteria to either select peaks, pink and blue, which are not too far, which are not overlapping too much, but also, more importantly, to discard peaks that we consider as background and not true origins. And this allowed us to exclude between 80 to 95 peaks. So that's really a lot. You can understand that in other models, when they do not do stranded library, interpretation are very difficult, repeatability, reproducibility is very low. So using that technique and that protocol, we were able to determine the origins. They are most located in the 5' UTRs. They are AT rich, and in fact, they are even done with a long stretch of A and long stretch of T. And there is also a sound motif implicated. We really wanted to be sure that the origins we selected with our protocol and with our approach are true origins. So we want to compare our data with already published data set. Origins haven't been published, haven't been characterized and cartographed in brucei, but there are some techniques that can give clues. For example, ChiP-seq, nucleosome occupancy, G4 structure, and marker frequency analysis. Anyway, we face a difficulty because all these techniques were performed in different reference genomes. So there was a very big bioinformatic works in order to translate positions in the different genomes. First, I wish to compare with ChiP-seq data. ChiP-seq, in fact, is a kind of ChiP-seq which download DNA RNA hybrid structures. So it was published in order to present some instability part of the genome, but we considered that it should download also origins, because origins are DNA RNA hybrids. And so here was the results are. So you also see many peaks. And we were very happy to find that we find more than 91% of our peaks in the ChiP-seq. So we have a very good reproducibility, concordance with that technique, which is quite different as SNS-seq. In terms of nucleosome occupancy, in the paper published by Murray, it was MNS-seq, and there was 4 replicas in BSF and PCF, and we really see a dip of nucleosome occupancy right over the origins with shoulder. That's what we see in blue in the bottom of the histogram. When we use a shuffled sequence, we don't see any enrichments. So that's what is presented on the right. I don't want to go into details in the G4 structure. It is still ongoing. I go through that because I don't think I have time, and I'm sorry to present all the stuff. This I didn't wanted to present, but one of I first wished to present you CRISPR-Cas because I spent lots of time to develop it. And as you mentioned, I was the first one who set it up in Leishmania, and I was very happy. We organized a workshop. We spread our plasmids all over the world as much as possible. But our technique was the first one, but was not very efficient. We moved to a second one, and now we are in the fourth one. Here, similarly, most of the work we did in the past was done on Leishmania measure, which was responsible for cutaneous leishmaniasis. And I was not that happy because I think it's more important to work on visceral leishmaniasis, which is a much more severe disease. And in addition, visceral leishmaniasis is endemic close to Montpellier, so I find it quite silly to work in major. But now we switch to Leishmania mexicana, which is also responsible for cutaneous leishmaniasis, and which is not endemic neither in Montpellier nor even in Tunisia, but only in South America. So I really want to set up a good Leishmania infantum strain for genome edition, and that's what I am preparing. It's a side project of my PhD student. I switched to another facet of our laboratory, which is molecular and cellular biology. And here also we want to decipher core cellular process. And this is a project that is supervised by Professor Patrick Bastien. So I want to give you the data we obtained in Phase7b, which is a kinesin of the flagellum attachment zone in Leishmania. The flagellar attachment zone of Phase was described in T. brucei, and it is all the zone because the flagellum is running along the cell body. So it's really a zone that is quite large. It has a key role in morphogenesis, in regulation of sync length, and in many things. There are 34 proteins with orthologs in Leishmania. And despite the fact that it is a much more discrete area in Leishmania, flagellum doesn't run all along the cell cycle, but emerge directly. So it's a very discrete zone, but we will to work on that. Doing immunofluorescence assay, comparing with other proteins that were already localized, we were able to localize Phase7b at the distal part of the flagellar pocket, and also to show that it is differently expressed in the promastigote and amastigote stages. We did the knockout, and we see a reduced growth. Both in the promastigote, much more severe in the amastigote. The reduced growth was restored when we do add back, but was not restored when we use for the add back a protein that was mutated at the kinesin domain. Oops, we are frozen. Oops, and after it goes too fast. Here I had movie, but I think that's why it was so heavy, because this was 3 movies, but I don't want to start. It's a pity because I appreciate this movie. Here is a parental, and we were able to record cells dividing during 18 hours. And you can imagine that it is quite difficult to record a moving cell during 18 hours. So we were able to record division, and with Phase7b depleted parasite, we saw impaired divisions, both in promastigote and mastigote. And that one divided quite normally, but in fact, division was not symmetrical at all. But that one gives birth to really monsters. When we look at cells in electron microscopy, we also see many cellular defects, abnormal cytoplasmic inclusions in the domain of the flagellar pocket, abnormal kinetoplast, abnormal basal bodies. We saw cells showing 6 transversal sections of the flagella in a single FP, and 2 other in another one. And in addition, by AFA, when we look at the other members of the flagellar pocket, such as Bilbo and FPC4, we see also many alterations. Here is a second movie, and that one is moving. So we were able to record the movement, and we used TrackMac, which is a plugin you can add in ImageG. Film is much more easier to do because they are very short movies. With TrackMac, we were able to make statistics and to show that depleted Fas7b parasites have an impaired motility. Then we send the parasite to a collaboration in the Czech Republic with Peter Wolf to infect phlebotomus, and we saw that development was reduced. And we also checked virulence in mice in a collaboration with Jeremy Mottram in UK, and here again we see a reduced virulence. So in total, Fas7b in Leishmania is required for cell morphogenesis, for cell division, and virulence in the mammalian host. Much more is presented in the paper. In particular, in addition to Fas7b, there is a homologue Fas7a, but it does not localize it localized to the basal body, and the deletion does not impair cell growth nor the cell cycle. Sorry for my dyslexia. Here I move to a third axis, which is led by MIVEGEC, and deals with a post-translational modification in trypanosomatid parasites, in particular the tubulin tyrosination. Tubulins are constituent of microtubules. Microtubules are important for core cellular processes, in many core cellular processes, in higher eukaryotes, and also in trypanosomatids. And in particular, the core set is made of microtubule, and it gives the shape of the cell. Here it's a TB, but it's the same for Leishmania. And also it's important in a stage of transition. Here are alpha and beta tubulin in humans. There are many isoforms, but what we're interested in is the tyrosination and detyrosination cycle of the tubulin in human. And in a first paper, Maud worked want to study. In human, deteriorization is performed by VASH protein, but it was complicated because there are two VASH proteins. So we did a collaboration to work in T-brucei because there is a single homologue. And in the first paper, we wanted to know what was the role of TB VASH in the trypanosomatids. So VASH is closer to VASH2 than VASH1. We did it by CRISPR-Cas, and we found that it was able to detyrosinate alpha and beta tubulin in trypanosomatid, and it alters growth and it alters the cell morphology. Here is a morphology of normal cells, and here you see how elongated it is when it is depleted. So this paper gives details on VASH in T-brucei. VASH in T-brucei was closer to VASH2 in humans, and it appears before the vertebral split in higher eukaryotes. Now she is interested in the role of VASH in Leishmania, and I will go there. She already obtained. I'm so sorry that we did not upload a good one. So in Leishmania, but what my message I want to give you now is that working in tyrosination, tubulinization in those divergent eukaryotes are interesting because they differ from those in humans. Both the alpha and beta tyrosin, both alpha and beta tubulin are tyrosinated in the trypanosomatid, whereas it's only the alpha in humans. So we expect that there is novel regulatory mechanisms, and that there is also recruitment of specific microtubule-associated proteins, and we expect to be able to determine drugs target. And she was founding, she was awarded ANR jeune chercheur very recently on that. Here is what she wants to do, to identify specific maps. So in order to do that, we were able to design tools, first to obtain a detyrogenated beta-tubulin T. brucei. Beta-tubulin is spread in tandem repeats in a single chromosome in T. brucei, and we were able to detyrogenate, to eliminate the codon encoding the tyrosine to each genes. It's a repeated gene, but we're able to do that. We used a ribonucleoprotein approach, Cas9 approach, which is very, very efficient. In the Sanger sequencing, you see that there is no anymore the triplets encoding the tyrosine. We also wanted to obtain a constitutively tyrosinated beta-tubulin. To do that, to impede VASH to remove the tyrosine, we modified the amino acid just before the tyrosine, and we also did the CRISPR, the same CRISPR technique, and we were able to obtain those parasites. In Leishmania, things are much more complicated because not only beta-tubulin is repeated genes, but it's also repeated in 3 different chromosomes. We were already able to delete tyrosine specifically in chromosome 21. That allele is responsible for tubulin in a mastigot form. But the other, we want also to obtain it, and it's currently ongoing. All these tools are coined in order to be able to do proximity-dependent biotin identification approach. And to do that, we fuse a turbo ID to an enzyme that binds to the tubulin tail in order to add or remove glutamine in the tyrosine tails. And we wish to be able to biotin, to label the map and to obtain it by immunoprecipitation and to identify them by LC-MS/MS. Here the movie is working, so I'm happy. Just I want to show you this picture to present you the fact that we have also produced antibodies and specific antibodies. And in the film on the left, you saw in green all the microtubules of the corset and those of the flagellum. I don't want to try again. But in red, you saw the mitotic spindle. And the mitotic spindle was labeled specifically with antibodies recognizing only detyrogenated tubulin, whereas in green it is an antibody that recognizes tyrogenated tubulin. We used expansion microscopy. That's why we see so many details. Really, I think we will learn so many things. I'm able to see in that picture so many things, so many details, some microtubules that were never described. Also, the nuclear morphology, we see things that we never saw in terms of DNA compactions. I'm very happy that now we have this technique in the lab, and I think we can learn a lot. So now I'm switching to the last part, which is much shorter. I am an MD in addition to be a PhD, and I work in a routine lab, as you say. We do lots of and even more and more PCR for routine dialysis, and I am responsible for the molecular biology pole of the National Reference Center for Toxoplasmosis, and I have quite a large time experience in comparison of method of molecular diagnosis in toxoplasmosis. But here I want to present a comparison I did in frame with the National Reference Center for Leishmaniasis, and I want to present you a comparison method. In the lab, despite or because we are a reference center, we are using a laboratory developed. Neither for Leishmania nor for Toxo, we are using methods we have developed, but we benefit from methods developed by others that were very good. But now we have to face the fact that there are kits that are developed by manufacturers. My first experience was in Leishmania, and the kit was really hopeful. It was I even forgot the name of that PCR technique, a NIC PCR. At the time, we were already doing a quantitative PCR, and sensitivity was half of the sensitivity we had. So it was an easy game to say that it was a very bad kit. Now I'm testing almost all kits that are distributed in France for toxoplasmosis, and I began with Leishmania. And here again, the game is quite easy. It's quite easy because the commercial kit has very poor sensitivity. We do almost the same protocols. We use serial deletion assay, which allowed us to determine performance scores that allowed us to determine the linearity zone and the PCR efficiency. And linear zone and PCR efficiency are good with the test. Agreement using a Bland-Hartmann test in considering the concordantly positive samples are good. We see a shift in the C.H.U value. The commercial center is delayed by a CP value of 5, which is understandable because there are less DNA targets. But this delta CP value is in frame with the total number, so the ratio of the targets. So really, this kit is well manufactured, but when you test it against a clinical cohort, what do you see? You don't need to take a very big cohort, but what do you see? You see that you miss 6 diagnoses. So really, this kit is finally optimized. However, it is less performant to detect low parasitemia than the laboratory developed. The method is not sensitive enough and can lead to false negative results when the parasite load is low. And in routine practice, a quarter of positive samples have a parasite load of less than 10 and cannot be detected by this kit. And this is probably, I say probably because I'm polite, but to the fact that the kit targets the rDNA, ribosomal DNA, while the reference method targets the kinetoplast DNA. So really, and all kits that are marketed now target that rDNA target. rDNA target is a very good target. It's used in many to target many pathogens. But in Leishmania, we have the chance to have kinetoplast DNA, which is much more repeated. So we shouldn't target that one. And I read this further development of commercial kits should take this into account. So I think it's a very different work for the basic research I do, but it's also important, and particularly when there are so big differences between reference method and marketed method. So I thank you for attention. I thank all my team. in particular the PhDs, postdocs, also collaborators in Montpellier in France, and also abroad, even in a Institut Pasteur. I can't thank her because I know she's not here today. Zina, you probably know her. And Arthur Schaff at the Pasteur Institute, who was my supervisor and still helps me when I need. And you see that I have many affiliations and also many funding agencies. Thank you. I'm ready for questions. I have an additional slide, sorry. This is, I told you that my lab has an old story with the Pasteur Institute in Tunis. This is a picture that was taken in 1970. I can say that I was not born. Almost, but not.
Thank you so much, Mr. Sterkers. I think this amazing presentation and this overview of the activities, the different activities that we have. So it's very interesting. So I'm gonna open the discussion. I have a few questions for you. So basically, I'm trying to do as you present as the same schedule. So I have a question about you observe any phenotypic expression difference in the aneuploidy parasites?
So this is hard to answer. I don't know what is a normal ploidy, so I don't know what to compare. We first identified a mosaic aneuploidy in a Leishmania major strains that were grown in culture for a very long time. And I think that's why we were able. It was because it was able to be 70% of triploid for chromosome 5 and 60% monosomic from chromosome 2. So it was easy to catch. But if you remember, in WGS, you saw other strands that were much more closer to disomy. So it's hard to answer. In order to answer your question, we looked in animals if the ploidy if the mosaic aneuploidy was maintained in some organs. We did that in the Pasteur Institute with Gerald Spate in mice, and we saw a mosaic aneuploidy, but it was hard to say if mosaic aneuploidy in liver was different in that one in spleen. That's what I say, we still don't know the role. We can speculate. It gives flexibility. And if you need a gene, you can increase the number of chromosomes. And this is not deleterious as it is usually in other systems. But we don't have evidence. Also for resistance, but for drug resistance, most of the time mechanism does not imply a chromosome copy number variation, but a copy number variation, but made of Leishmania is very able to keep small circular DNA, and for multifusin resistant, for example, it's like that. But yes, adaptation.
So, and in the way concerning the study, you said that you studied the Leishmania major, Leishmania infant, Leishmania deciphering, and we know there is a difference in the chromosome number. So, any difficulties between them, at the cellular degree?
As regard the measure and infantum, they both have 36. Mexicana have 35, that's true. It has 35 because there is a fusion event. So the only difficulty is when you work in the bigger one, not to make a mistake. But no, it's not a big deal.
Yes, and finally, maybe it's the most interesting for me. It's about diagnosis. So basically, when you do your own in that diagnosis. So you do it like specific for species or just for the generic one for all the species. And my second question, did you try to set up the isothermal molecular isothermal techniques like the isothermal amplification?
I will answer the first one because I will forget the second one anyway, and I will ask you to repeat the second one. Yeah. So of course, we need identification. In particular, because in France, cutaneous leishmaniasis is almost not endemic at all. There are few cases of cutaneous leishmaniasis due to infantum. But in fact, most of cases of cutaneous leishmaniasis come from abroad. Abroad means from here, from all the South Mediterranean Sea border countries. And it's important to know if it is major tropica, any species. So it's very important. We have to. But in addition, we also receive cases from Americas, in particular French Guiana. So we really need, in that case, we really need to, it's even more important than for the so-called old world leishmaniasis, because some of the species from South America can lead mucocutaneous. So to be short, we have a pan-Leishmania PCR, which is able to diagnose all the Leishmania species. If we work on a blood sample, we stop then because it's only Leishmania infantum donovani complex, and we cannot discriminate between those two. They are not different species, but a complex. But if it is a cutaneous form, yes, we have to go other set of PCRs, and we are still using on clinical samples. We are still using Sanger sequencing.
So for the identification, you use sequencing.
Yeah, we still use Sanger.
And the target is KDNA for sequencing.
Yes.
The target is KDNA.
We sequence specific genes because it goes deeper in discriminating the species. If we cannot, yes, we sequence ribosomal DNA gene, but we cannot go as deep. And sometimes we can just say old world, new world. So we are not. But if the parasite burden is low, single genes can be negative, and RDNA genes is already a clue.
Yeah, so my last question is about the blood of isothermal, molecular isothermal.
This I have no experience in Leishmania. I have almost no experience. I've tested one in Toxoplasma. Now we are using it for malaria diagnosis. Isothermal identification is not different from PCR, but the manufacturers was very, very clever in 2 points of view. First, they say it's much easier in developing countries to do isothermal because you don't need a thermocycler. This is almost not true because if you have electricity, you can have a thermocycler. But it was one way to make lots of isothermal identification. And in France, they were also clever because they say, no, for isothermal amplification, you don't need to be a proficient molecular biologist. Anyone can do it in any desk. So now we do it. We don't do it in the molecular biology lab, but on a bench like that. But in Leishmania, I know it's an option, but we're not using it. Yeah, and you can ask in French. I will also understand.
My question is, if you use the Leishmania
If I've used phase as a vaccine, the phase 7 and the... no. In fact, as you have probably understood, I am more in really deciphering the core cellular process. Of course, phase 7 and also VASH depleted parasites are virulent. So in that respect, they could be a vaccine. But I don't think that a vaccine would be done with a virulent strain anymore. It's not possible. Maybe for dogs in France, we are interested in vaccine so we have one for our hunting dogs. We care more of our hunting dogs than our children, because we have a vaccine for the hunting dogs, but we don't have a vaccine for the children, despite the name, which is Leishmania infantum and not Leishmania caninum. And but you are right. It's I know that some. I think it was Centrin depleted has been used, so it could be. It could be. If you are interested, I can share the strain, but they are manipulated with Cas9. But now, the one I've generated, so now really we can obtain Leishmania depleted parasite with almost no scare in the genome, no scar in the genome. You want to delete the gene, you delete the gene. I want to eliminate a tyrosine in the genome. I just eliminate the tyrosine. I don't add drug markers. The Cas9 I use is in vitro synthesized, so after 2 or 3 half-life, there is no Cas9 anymore in the Leishmania. Guides are RNA molecules that have also half-life, and donor DNA similarly. So